Yeah, so I'm Alf Wachsmann. I'm the CIO of the Max Delbrück Center for Molecular Medicine. And I will tell you a little bit about the science we are doing, and then the new cluster we put in place very recently with DDN technology, and show you some very first results that we got from our first test users on the system. So first of all, about myself, I lived and worked in the US for quite a while, where I put in my first DDN system. Then I moved to Japan, where I put in my second DDN system, and I'm now back in Germany, where I put in my third DDN system. I'm still not getting paid by DDN, by the way. But I really like the system because the cost of ownership is just really great. So you put the system in, we have the embedded version of the grid scaler. It all comes, you plug it in, and you have nothing to do except change the disk once in a while. So it's just great. So my institute, the Max Delbrück Center for Molecular Medicine, is part of the German Helmholtz Association with 17 other institutes, and we work in the area of medicine. And I'm talking a little bit at the beginning about our institutes so that you know who we are, where we are. We are located at the outskirts of Berlin in Germany, at the northeast corner. We see its beautiful green campus here. This is all kind of us. We are together with a biotech park where a lot of startups, many of them out of our institute, are. Then there's another research institute for pharmacological research, independent of the MBC, and then there is a joint venture between us and the Charité University Hospital in Berlin, where we do experimental clinical research center, is ECRC. So it's a great mix of really basic research, clinical research, and commercial applications of the research in one big campus. So it's a very nice atmosphere in the area to do work. So our institute is technically only 20 years old, 22 years old, but the research that was put into our institute goes back some 100 years already. And then through the GDR, there always, so East Germany, there were 4 or 5 research institutes that then after the fall of the war were all merged together into the Max Delbrück Center. So the whole research history goes much further back than these 20 years that our institute exists. Now, the mission statement for our institute is to investigate basic biological concepts important for the understanding of disease mechanisms and to overcome the traditional gap between basic biomedical and clinical research. And I'll come back to that a little later, this gap where it takes like 20 years from drug discovery or discovering the mechanism how in biology something works to having really a drug on the market. It's just too long, 20, 30 years. And this is part of our mission to close that gap, which is why the research campus in Berlin is as it is, with the startup companies, our institute, and then the clinical research in ECRC to really make everything located close together. Now, in numbers, our institute has a budget of roughly 100 million euros. The 76 million euros is from the German government, so that's kind of steady funding, and the rest is third-party funding. We are roughly 1,600, 1,700 people on site, so it's not a small institute, and that does not even include the guests and postdocs and so on. So we are just under 2,000 people on site. Yeah, we do great science, some published papers, some with really big impact factor. But yeah, what science do we do? So essentially there are 3 areas of research that we do. All of this is based on the cell and molecular biology of the human body, and we use, of course, mouse models and other models to do this. We look at signaling pathways, developmental biology. So one great project there is how to make pancreas cells out of liver cells. so that when you have pancreas cancer, so the goal is to just extract some liver cells, make them into pancreas cells and reimplant them. We are not there yet because we don't really know how these cells differentiate in the human body, but this is the kind of research we do. And so the three big areas we're working on are cardiovascular, metabolic diseases, so from heart attacks all the way to blood flow problems, blood clotting, and so on. Then cancer, sure everybody does cancer, so do we. Then diseases of the nervous system, so we investigate all sorts of neurodegenerative diseases, Alzheimer's, Parkinson's, multiple sclerosis, and all these things that are really plaguing mankind. And then more across these three research areas is the medical systems biology. This is more a concept how to do research than really a research area in and by itself. Systems biology and systems medicine is more the holistic view of things, where we understand now that, for instance, if you have a single mutation in your gene that does not cause a single disease, but one mutation can have multiple diseases as a consequence. And also, one disease does not necessarily map to one problem in your genome or transcriptome or whatever, but it can have multiple causes, in that sense that multiple things have to go wrong before you get this one disease. And this holistic view of these many-to-many relationships in causes to results then, that's called systems biology or systems medicine. And that area is where, for instance, a lot of bioinformatics people are working, where they really try to model, mathematically model, and then simulate in the computer what's going on. So this pathway signaling, we have whole networks of which protein transforms into what other protein, which affects this and that and the other things. That's exactly what systems biology is all about. Okay, that's what we do. And now let me present to you the new compute cluster, so the compute resources that we are now using to do this kind of research. We had a cluster in place when I arrived at the lab that's now 7 years old, so somehow they skipped one generation of investment, probably because no one knew what they were doing. Was some 100 compute nodes with an NFS file server for the data. And everybody who is using NFS in a cluster knows that's, of course, not a high-performance file system. And the researchers were seeing exactly all the problems you would expect. So if one user runs 2 jobs, then the file system would slow down, that no one else could essentially do work. And so we now purchased a new cluster, which happens to be in a European tender. Dell won this bid. So we have some 150 compute nodes in 3 different specifications, and I'm not going through all of them. The important part is that each chassis here has 2 10 gigabit uplink into the cluster. The blades themselves, so each individual node has only a 1 gigabit connection. So we are not getting 10 gigabit all the way to the compute nodes. It's only 1 gigabit. And then the DDN part, it's a SFA 12K E20, so it's an embedded 12K. We're using GPFS as the file system. And so the metadata is on these 3 times 300 gigabyte SAS drives inside the DDN system. And then the rest, it's 80 terabyte raw, it's 630 terabyte usable GPFS space to the cluster. And yeah, we do have an export facility that bridges GPFS and to NFS and CIFS. So if someone from the desktop or from some visualization workstation wants to access it, they don't need a GPFS client. They can just use one of these other protocols. The cluster itself, all cluster nodes do speak native GPFS. And then the old cluster, some of the newer NFS file servers, we connected via a 10 gigabit link to the new cluster. So the benchmarks I'm going to show you now are all done on the new compute nodes. And when I'm talking about NFS, it goes via this 10 gigabit link to the old legacy NFS file servers, and the GPFS network file system is sent using the new DDN hardware. But the clients are the same. Again, remember they have only a 1 gigabit link into the cluster, but the file servers are connected 10 gigabit. And we haven't had time to do full-scale benchmarking testing, but these are very preliminary numbers just to see what this new file server infrastructure can do for our science. So there is no scale-out testing for how many clients can I use to make the grid scaler go to its knees. So the tests I've done we have done from a single client only. But the numbers are really promising. So not changing anything at all, just using the different file server and file system. We get speed up here for this particular application by roughly a third. Again, this is without doing anything. So the problem here is gene sequencing application. When these gene sequencing files come out of the gene sequencing machines, they have to be assembled, and then after the assembly, they need to be sorted and indexed so that the tools further down the pipeline can access things more quickly, that you can find the locations on the genome more easily. And the step here is this sorting. The parameters, SAMTools is an open source tool that's widely used for this step. And the parameters we're using in this particular benchmark here are from the old cluster. So this is not adapting to the new cluster yet, where we have 16 cores per node. The old cluster had only 8 cores. That's why the threading here is still 8 cores. We really wanted to have an exact comparison. What are we doing on the old cluster to what are we doing on the new nodes? And the working set, so the in-memory space we are getting for this application is only 8 gigabyte. The new nodes have 128 gigabytes. So we could crank this up quite a bit, and in fact I did that, and I've now got these numbers down to 8 minutes. But that wouldn't be comparing apples to apples, right? This should be in comparison just for the file system infrastructure. And without, again, changing anything at all, we already get this speed up. And this is something here on a 20 gigabyte BAM file. These BAM files, so the gene sequencing files, range from anywhere from 6 gigabytes up to 60 or even 100 gigabytes. So this is more on the smallish side for a BAM file. So we expect for real production runs to make a really significant difference for our production. Now the second application is a little bit more tricky. This is variant calling with the Genome Analysis Toolkit, GATK, from the Broad Institute. The version of GATK we are using here is version 1. something. The GATK right now is already at version 3. So there are major performance increases we can get by just upgrading the version of the GATK. But there are some scientific and other reasons why we are still at this old version. And we are working right now with the scientists to work through all the various reasons, mostly scientific reasons why we are still on the old version, to upgrade this to the latest version. So don't take the absolute numbers of runtime here, but more the relative, the difference. Again, we wanted in this comparison really compare apples to apples. So the problem is that there are 2 different versions of this variant calling in the GATK. One is that you have one reference genome and one genome where you want to find all the mutations compared to that reference genome. Because these mutations are what you're looking at then to identify diseases or mechanisms for diseases. So that's one version. You have a reference genome and one genome you're interested in. That's not what we do. This version of the variant calling is using for statistical reasons one reference genome, but in this case some 250 genomes we are interested in finding the mutations. And the reasons why this is important is that the statistics get better when you use more files. So it's a slightly different algorithm you use. The gene sequencing machines themselves produce errors. And These errors are, of course, not mutations that the patient or the mouse has. So with using many more files for this comparison to the reference genome, you can even out these statistical errors that the machines themselves give us. And so this problem here, 250 BAM files, each of these files has, in our case here, 6 gigabytes up to 40 gigabytes of size. So you can do the math, that's some 5 terabyte working set we're talking about. The speed up we're getting, again, without tweaking anything else, right? The same working memory size, same threading. On the NFS server, if the program finished at all, it took about 36 hours. But if someone else was already putting some note on the NFS server, it was typical that these essentially never completed. For reasons I don't quite understand, this problem was never that important to run at our institute, so this wasn't a big problem. However, going forward, we have built with the Charité University Hospital a new institute, the Berlin Institute of Health, where exactly this program will become routine. So we want to do this going forward essentially on a routine basis. So that's why it's so important that now with, again, just GPFS and the DDN hardware underneath it, we get a speed up that is enormous. We are now down to 5 hours without changing anything at all. Again, the same clients, the same problem, just the file system is different. And again, we know that upgrading the toolkit itself, we will get another, I don't know what the factor will be, but significant speed up. And that is a qualitative difference in runtime. So imagine a researcher submitting this batch job and having to wait a day and a half for the results. He already forgets what he was trying to do when he submitted the job once he gets the result. Getting this down to 5 hours now, that's easily within one workday. So if a researcher submits this in the morning, he has the results before he goes home and can do planning for the next day. Or he goes home, submits this job before he goes home, and when he comes back in the morning, he has the results. So this alone makes a huge difference for the way we will do research going forward. Then, as said, NFS, if the job completes at all. Now with GPFS and DDN, we can run multiple of these jobs in parallel on the cluster. I mean, yeah, we have the compute nodes, we had those before, but it doesn't matter if the NFS server can't serve the files. Here with GPFS, and again, I have not done the scale out test, so I don't know how many clients I have to run this application on for us to see any performance difference. But I'm sure we can run many of these applications in parallel on this kind of hardware, on the grid scaler, before we see any kind of performance degradation. So that also, moving forward with this application in this new institute, will make a huge impact on the research. So the quality of research will be much, much higher, much, much better. So this is some very preliminary testing with some nice users. The main system administrator is right now on vacation. Once he's back in 2 weeks, I expect this new system, the cluster and the storage, to go into production, so that then not just the gene sequencing people, but really everybody gets a shot at it. I talked about the bioinformatics people. They write their own application. I have no idea what they're doing with the cluster on the file system, so I'm excited to see that. Everybody's looking forward to using this new hardware because these numbers already made the round through the institute, so everybody's eager to get their hands on the new hardware. And so we are looking forward to really putting this to real tests. And then, of course, moving forward for the new Berlin Institute of Health, I am already planning for a new DDN system where we have a new gene sequencing facility where we expect this to be really the production pipeline where the clinical researchers put in routinely probes, so samples from the patients through this system, where it really is important that doctors get the results within a workday, for instance, or overnight. Because, well, a week later we have the results and, oops, sorry, the patient is dead. So doesn't help, right? So it is becoming very critical that this kind of application runs really quickly. And so with that, I want to thank Martin Siegert, who is the HPC system administrator on the system, who put in a lot of work to make this all work together with folks from DDN. Tommaso sits here and was very instrumental. And then Bastian Schröder from the bioinformatics side, from the gene sequencing, who supplied the BAM files and the application and run the tests. So this is our beautiful campus. Come visit us. We are an open lab. Thank you very much.